Indian writings from 1500 BC call it the king of diseases. It can be found in the writings of Aristotle and Plato. It has even affected U.S. presidents like George Washington and John F. Kennedy. It's been around for thousands of years, yet we are still looking for medicines to treat this disease. Malaria is caused by a parasite that kills 500,000 people each year, and it all starts with a mosquito bite. The parasite is called Plasmodium Falciparum, and it's like Houdini at evading the immune system. Infected red blood cells will express a protein called Plasmodium Falciparum erythrocyte membrane protein 1, or PFEMP1. This protein allows infected red blood cells to bind to host cell receptors, as well as uninfected red blood cells, causing sequestration and rosetting. This allows the parasite to escape clearance by the spleen and can cause blockages in the microvasculature, leading to inflammation, organ failure, and death. Mortality rates of malaria patients are highest in the first 24 hours after hospital admissions. Commonly used anti-malarials do not significantly reduce cytoadhesion parasites within this time frame. Identification of anti-adhesion molecules may improve patient outcomes. Our lab has designed a method to efficiently identify anti-adhesion compounds. Torrey Pines Institute for Molecular Studies has created 80 libraries containing a total of 30 million compounds. Compounds are tested for inhibition of host receptors binding to malarial receptor binding proteins, PFEMP1s, that are expressed and immobilized on bioplex beads. Testing of binding in the presence of compounds is tested using a fluorescence tag. 2 compounds have been identified using bioplex. Both of these compounds have demonstrated inhibition of binding of 50% at concentrations down to 250 nanomolars. Bioplex results have also been corroborated using live infected erythrocytes, and both compounds have demonstrated inhibition of binding of 70% at 250 nanomolars. In addition, these compounds have also been tested for cytotoxicity. Both compounds have demonstrated no cytotoxicity at concentrations up to 20 times of the IC50. This is very important in providing future directions in making a potential drug candidate.